So, good afternoon, everyone. Good afternoon, all the guests here. Today I'm going to speak about metabolomics and microbiome in disease pathogenesis. So, before I delve into the into my topic, I would like to tell you how I ended up into this topic, or you can say the crosstalk between metabolomics and microbiome. Uh, how metabolomics has happened to me is when I did my PhD in metabolic networks in human energy metabolism, and that's how I got introduced into metabolomics and metabolic data and metabolic networks. And how microbiome happened to me is microbiology was always my microbes are always my passion. Like, no, I always wanted to work on microbes. Uh so after few years of my after my PhD, so I delved into the area of microbiome or microbiology, and started working on various diseases and their pathogenesis. And then I brought my expertise of metabolomics uh, into the area of microbiology. So that's how this topic has come to my mind when I was being invited, that what topic I would be speaking in this talk. So it's metabolomics. So it's basically the crosstalk between metabolomics and microbiome in various disease pathogenesis. So I'm Dr. Abhishek Sengupta, and I'll be guiding you through this exciting journey of understanding how these intricate components, basically the components in metabolomics and the components in microbiome, uh, intersect among each other to unravel the mysteries of various diseases. So let's get into it. So, today, what we are exploring is the fascinating realms of metabolomics, microbiome, and their pivotal role in disease pathogenesis. So, in this area of precision medicine, delving deep into molecular interactions is crucial for understanding complex diseases. The fusion of cutting-edge bioinformatics with these fields is instrumental in extracting meaningful um, insights from vast data sets. So let's begin by exploring the few fundamentals that we need to know before getting into this juncture of uh, metabolomics and microbiome. So metabolomics. So our starting point is metabolomics. So what is metabolomics? It is the meticulous study of small molecules or metabolites present within the biological system. Right. So whether a cell, whether a tissue, or blood, or any biological sample, so all these small molecules or metabolites that are present comprises the metabolome. So think of it as a molecular snapshot, capturing the metabolic state of, or the present metabolic state of cells or organisms. These metabolites, ranging from simple molecules to complex compounds, are the building blocks of life processes. So by deciphering these patterns, we gain insights into disease states and metabolic dysregulations. So whatever dysregulations that we see, or in a cell, or in a diseased or in a disease state, or in a healthy condition, can be understood by these various states of the metabolites. Now let's shift our focus to our next area, that I mentioned in my title, that is microbiome. How my metabolome and microbiome crosstalk among each other. So now let's define microbiome. What do you understand by microbiome for the people who are who have I'm sure everybody has heard about microbiome these days, because that's the buzzword, one of the buzzword in precision medicine, or in personalized medicine these days. So, or in microbiology. So, let me define what is microbiome. Microbiome is a dynamic community of microorganisms residing within us. So, this complex ecosystem that is found in our gut, skin, or various parts of our body, and various plays a vital role in health maintenance. It influences digestion, it influences immune responses, and even the impacts diseases beyond its immediate environment. Right. So, understanding the uh, intricate this these interaction between the host cell and microbiota unveils new avenues for disease exploration. So, it opens up various avenues of understanding the disease, understanding how the host cell, or how the microbiota interact among each other, right? So, that can be understood through this field of microbiome. So you can see here in the picture, like how we have good bacteria, we have bad bacterias, and how it comprises the whole uh, 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 the whole microflora of our body. Now, what do we understand by disease pathogenesis? Since I'm going to talk from the perspective of disease pathogenesis, like how we can understand disease pathogenesis, or how we can provide solutions to various diseases through this interaction, or through this crosstalk between metabolomics and microbiomics, or metabolome and microbiome, is exactly what I'm talking today, or I'm going to like no deal about today. So, disease pathogenesis, it is a multifaceted approach. So disease pathogenesis is a symphony is of genetic predispositions, environmental factors, and now microbial influences also. We are realizing that our microbiome contributes significantly to our health, and in some cases to our illness also, right? So it's like a puzzle, right? Where the microbiome's role is essential in understanding the complete picture, whether you are healthy, whether you are you are diseased. Everything is is more or less being judged by the microbial composition, because it has been reported that uh, that we have millions and millions of more microbes than the number of cells that we have in our whole body. So that itself gives us a picture that how microbes has a greater role in causing a disease or keeping us healthy, right? So integrating metabolomics and microbiome research is like adding more pieces to the puzzle. So I was talking about this puzzle. So if we integrate this metabolomics and microbiome research, that means we are adding more information. So though this is quite complicated and quite data intensive, or data uh, dependent process, but still it enriches our understanding of the disease pathogenesis. Now, let me first tell you how do we analyze my metabolic. Though my previous speakers have spoken about a few different methodologies or different experimental methods by which we can do metabolomics data. So as we dissect metabolomics data, imagine it as a treasure trove of molecular informations, right? So techniques like mass spectrometry and nuclear magnetic resonance help us in identifying and quantify metabolites. So that we all know. Like, no, I'm sure that you have already listened to some of the speakers about how do we quantify these metabolites, right? So through a meticulous process of data acquisition, pre-processing, and normalization, and convert raw data into valuable insights. However, this journey isn't without challenges, right? Data complexity, for example, as I was telling you, that data complexity, dimensionality of the data, and identifying unknown compounds are hurdles that we must overcome. So on the left, you can see here on the left side of this picture is a mass spec setup. Here you can see the sample preparation, then ionization, where the molecules are getting converted into ions for analysis, then there is a separation based on their mass to charge ratios, detection. So that there is a detector, you can see here, measure ion abundances to generate a mass spectrum for molecules. Now, on the right hand side, you can see certain graphs, certain like no these peaks, what we call them as peaks. These are basically mass to charge graphs. This this whole picture is basically called as mass to charge MZ uh, charge graphs. So what it displays, it displays ions abundance as a function of their mass to charge ratio, providing information on the molecular composition and structure of compounds in the sample. So these peaks, you can see these peaks and their values. Peaks represent the ions with specific MZ values, and their intensities indicate relevant abundances, aiding in identifying and quantifying the various substances that are present in a particular biosample, right? Sorry. Okay. Meanwhile, the world now, again, I'm just like no hopping in and hopping out of metabolomics to microbiomics. Now again, I'm here in microbiome data. So now just now I have talked about how metabolome data is collected and analyzed. Now here, this is actually a part of the work that has been done in my lab. So the world of microbiome analysis takes us to realms that were once hidden. So techniques like 16S RNA, rRNA sequencing and metagenomic reveals the microbial diversity within the ecosystem. So taxonomic profiling. So what we do is taxonomic profiling and functional predictions help us to understand which organisms are present and what roles and functions they play. With these tools, we unveil the microbial citizens, that is the microbial the t- t- ta- taxas that are uh, present and what roles and functions they play. So So this graph from one of my so this is from one of our publications, so from our lab, where in the vaginal microbiome sample, so what we have done, we have taken a vaginal microbiome sample, and it was analyzed to reveal the microbial composition as well as the pathway expression levels in term birth, right? That is so the and PTB samples. So what we have taken samples from we have taken samples in preterm birth, like no where we see preterm birth, right? So these are basically called as preterm birth samples from vaginal swabs, right? So from there we have uh, the profile, we have done the taxonomic profiling and functional predictions of the different microbes. So we observed an increase in the abundance of the pathogenic microbes and overexpression of disease associated pathways in the PTB samples. So PTB is again preterm birth samples. So we have seen some changes in the microbial expression microbes and the type of microbes and their overexpressions. Similarly, data from the gut microbiome, skin microbiome, and more can also be analyzed similarly to understand and explore their contributions towards any disease condition. So this is the story of how we analyze the microbiome data. Now the real magic happens. So here I'm just uh, getting into a picture which I wanted to convey to all of you, is the integration of metabolomics and microbiome research. So the real magic happens when we bridge the gap between metabolomics and microbiome data. So micro bioinformatics enters at this stage to guide us, because there are various bioinformatic approaches, there are various ML based algorithms which actually help us in integrating such two bigger areas or massive areas like metabolomics and microbiomics, right? So by integrating these data sets, we build a more complete understanding of disease mechanisms. Think of it as a solving a multi-dimensional puzzle. Again, I was talking about that puzzle where statistical methods and machine learning algorithms helps us in identifying different patterns, biomarkers, and predictive models. So imagine the intricate metabolic and microbial interactions. So within our bodies as networks, right? So I'm just depicting here the interaction between the various metabolites and the various and the different microbial taxes into networks, right? So the connections that form the various connections that form the basis of life processes. So network analysis is our magnifying glass. So it's sort of our magnifying glass into these intricate connections, right? So we do a lot of network analysis to determine those connections between the microbes and the various metabolites, either in the host or the that are produced from the microbes. So with it, we uncover key pathways, identify essential players, and visualize the complex web of molecular interplay. The visualizations, this type of visualizations help us navigate these networks, giving us a clearer understanding of their significance. On the left hand side, you can see here a sort of a picture. Many people must be thinking it is a picture of it is a network taken from string, but not it is not taken from string, it is taken from stitch database, so which is a metabolic network based database. It's so it shows how metabolites interact with one another, right? So the database showing so similarly on the right hand side, the you can see the microbial so again this has been taken from one of our research paper, where we have shown the microbial interaction networks we developed in our lab. So these nodes that you can see here are mostly the microbes that are highly abundant or that are highly present in the samples, right? So so these microbial interaction networks was developed by certain softwares in our lab, which shows how different microbes of the human microbiome interact with each other. Network analysis further, so this type of network analysis further allows us to integrate the interaction between the microbes and the metabolites, which can be further studied to understand the pathogenesis of a disease. So pathogenesis of for understanding of pathogenesis of disease, we need to integrate both of them in a much more detailed way. So the more we integrate, the more complex the networks become. Now, I'll show you a very useful slide, which will show you the workflow of how we can integrate a metabolome and a microbiome based study, right? So here the first is the study design and sample collection. That is very much a necessary step. So definitely we need to design a study and sample collection, and then we do the data acquisition. So we perform metabolomic analysis using techniques like mass spec, NMR, sequence microbiome methods like 16S rRNA, shotgun sequencing, etc. So both type of data acquisition will happen both in microbiome and metabolome. Then there is data pre-processing. Now data pre-processing again will happen for both the types of data, right? Then feature extraction. So identifying metabolites and their corresponding intensities in metabolomic data is what is our aim, and then assign taxonomic identities to microbial sequences and quantifying their abundances, right? Then statistical analysis. Statistical analysis is performing statistical tests to identify differentially abundant metabolites and microbial tests. So now slowly we are integrating the metabolomic data with the microbiome data, right? So here by using statistical analysis we can we can easily do some sort of sort of relationship between the metabolites and the microbial taxes. Then we do the real integration, where we map metabolites to metabolic pathways and identify the potential interactions with the microbial functions. Use of statistical methods and different bioinformatic tools to identify correlations between metabolites and the microbiota. Now, next, I have already spoken about it, that like how we do network analysis, and then we can do the network analysis. So that was a quite a futuristic thing that we have not done as such yet, but it is still ongoing in our lab. That where we are integrating the microbiome data with the metabolomic data, right? So this is basically where we do the network analysis to understand the integrations. Then biomarker discovery. So this is basically again the outcome of of this integration, that we identify potential biomarkers associated with the studied conditions. Then pathway analysis. This is a very routine work, like how we do the pathway analysis, interpret metabolic pathways influenced by altered metabolites. So if there are any altered metabolites and microbial taxes, then then we assess the potential impact on disease pathogenesis, and then you can proceed with the visualization. So this we do with every bioinformatics work, where we visualize such analysis in the form of certain visualizations, and definitely we can go for validations if we have resources with us.
A kind reminder, Professor Sengupta, to the time is
Yeah, I'll just
exceeded.
quickly.
Thank you.
Yeah, sorry. So now I will quickly go through some of the applications. So I hope the audience have understood. This is my main aim is to make you understand um, the uh, the this integration between metabolomic and the or crosstalk between metabolome and the microbiome. Right now, there are certain applications people have worked on it. Many people have, like, I guess I'm sure there are one or two talks where people will talk about some there is one speaker who is going to talk about diabetes, type 2 diabetes. I guess I think it's in this session itself, the impact of COVID-19 and oral microbiome of type 2 diabetes patterns. So this is very one of the best applications that I have just spoken about. So biomarker discovery, therapeutic insights are the two important applications of this type of integrations. Now there are challenges are like no of course there are certain challenges, the variability in sample collection, experimental protocols and data formats. So all these are various challenges that we face, or we will face while integrating such huge data sets like metabolomics data sets and microbiome data sets. Now this is not exactly application, this is some case studies that people have done. One is this the disease this recent studies that have shown individuals with cardiovascular disease exhibit elevated levels of a specific microbial derived metabolite called trimethylene n oxide, which is shortly called as TMAO. And this has actually, these again acts as a one of the biomarkers to show the this the as one in cardiovascular disease, right? Then in diabetes mellitus, it has been seen that there are elevated levels of branch chain amino acids like BCAAs. You are quite aware of BCAAs. What are BCAAs? So this shows as one of the best biomarkers, like no to identify or to detect, not identify, to detect diabetes mellitus. Next is cancer. Now, in cancer also we have seen the role of microbiomes or microbial taxes, like no which have like for example notably fusobacterium nucleatum has been associated with colorectal cancer. So this is one of the best example that I can give, how microbiome is related to the metabolome or metabolomics in cancer. Now neurological disorders has I'll not get into it. So you are all aware of the gut brain axis barrier, and how microbes modulation on this onset and progression of these disorders, like especially Alzheimer's disease, or like no the various other related disorders. Then obesity also has a lot of has been seen a lot of relationships. So you can see these amino acid biosynthetic pathways and their partial and presence, and their vitamin biosynthetic pathways, and all these are in bacteria, and how they are related to the pathogenesis of obesity in human has been seen in various papers. Now in why to leave autoimmune diseases. So autoimmune diseases also has a huge role of like the huge role of microbiota in autoimmune diseases also has been observed. So you can see these are the results that has been taken from. So this is galinarium in the liver biopsies of autoimmune patients, that has been seen that how the gut barrier gut barrier function, and this particular microorganism has been seen a lot of relationships. Respiratory diseases also there is. This is going to be presented by one of our another speaker, that is COVID-19 and oral microbiome. So they will uh, cover a lot of these such relationships, right? So liver diseases has a lot of relationship, like in NAFLD has there is a lot of relationships between uh, microbes and these diseases. Like skin diseases also has a huge role of skin microbiome, and the the uh, the metabolome, there is a lot of relationship between them,
Dr.
right?
Sengupta, if you would come to a conclusion, a bit,
Yeah.
you're quite a bit behind, 10
So,
minutes.
so, so, this is my last slide. So
Mm.
what are the future directions? Like future directions, it emphasize the incredible potential of integrating metabolomics, microbiome research, and bioinformatics to actually unveil the intricate interactions underlying a wide range of diseases. As researchers, we continue to explore these connections. We move closer to a deeper understanding of disease pathogenesis and the development of personalized approaches to diagnosis and treatment. So, as we glimpse into the future, it is clear that journey of metabolomics, microbiome research, and bioinformatics is just beginning. With advancement in technology and multiomics integration, we are on the brink of revolutionary discoveries. These insights will definitely transform our understanding of health and disease, shaping the future of medicine itself. So this is my team, at Amity University. We have these our collaborators. This is our lab, and so I have already concluded. These are some references. Uh, so I'm open to questions.